So welcome to today's webinar, Programmatic Access to UniProt using Python. We're going to do this as a bit of a half and half webinar. I'll be walking you through the examples and how to find data on our new website. And my colleague will be walking you through how to use Python to access the same data and even go into more detail at how to access that data. So my name's Emily Bowler-Barnett. I am a scientific curator at UniProt. And a lot of the manually curated data that you find on our website is what curators like myself put in there. So we read the paper and the scientific literature that's around at the moment, and we put that data into the UniProt format and onto our database and website.
And I'm Aurélien Luciani, and I'm a developer in the UniProt team. So I work more on the development of the website and different services at UniProt. And today I'm going to walk you through the code examples in order to access the data that we provide through UniProt.
So for those of you not aware, although I'm sure most of you are, UniProt has a mission to support biomedical research by providing a resource of freely available, stable, comprehensive, and accurately annotated protein sequence and functional information. This includes amino acid sequences, both of canonical forms and isoforms of proteins, including naturally occurring disease variants that cause disease. It also includes data on physiological function, subcellular localization, pathways that protein's involved in, interactions, whether it's involved in disease or not, and if we know what variants cause that. We also have structural information, including topology and 3D structures where appropriate. And we also host data analysis tools for our users, such as alignment, blasts, and one we're going to show you today, which is retrieve ID mapping. We have over 1 million SwissProt proteins, which basically means they've been manually annotated by curators such as myself, and over 200 million Tremble entries, which have been automatically annotated by rule-based and curator-led rules for the data. So today's aims and outcomes is to help you understand how to access our data, both on the website and programmatically, and how best to pick an option for the workflow that suits you. Hopefully, you'll be a bit more familiar with how to use UniProt and its services and what we can provide you with, and where to find the relevant help and documentation to aid that access to our information. And we're also going to show you how to use Python and give you some example scripts to retrieve protein entries for specific fields, and also filter within them results for your annotation type and potentially protein character interest that you might be interested in.
So today, what we're going to show you is mainly the API, but this is only one of the multiple ways to get access to UniProt data. And it depends a bit on what you aim to retrieve. So we have multiple options to retrieve the data. If you want a big download of the whole UniProt data, you would be more interested in the FTP way of accessing the data. So you can find the FTP on the link on our website. So there you would be able to retrieve the whole of UniProt data, and probably that would be for anyone that needs really everything, and then that would be willing to do some post-processing really to extract whatever they're interested in. On the other side, we also have the website download. So that would be more for smaller downloads. Anything that would be just a one-off, you just need that information that one time, and it's fairly small. And so you could just go on the website, browse the website, and you will see in most of the pages, you have a way to download the data that you see on the page with this download button. And right in the middle, that's where we have the API. So API is more for medium-sized download. You don't want to download the whole thing. You don't want to download the whole of UniProt through the API. It's not what it's meant to be used for. And so it's more if you have a specific query for a subset of the data that you're interested in. You can also customize the download. That will show you that today. And that could be either for one-off download, or that could be also to integrate in workflows, in scripts, anything that, for example, at each new release, you would need to download new data, then you could use the API for that. Just for a bit of context, so the new website has been released in July 2022. So you can browse it now at www.uniprot.org. And the new API has been created at the same time. So the new website API has been used to find a way to provide the data to the website and also at the same time to provide the data for programmatic users. So it means that in a nutshell, that everything you see on the website, you can be sure that you will have a way to retrieve that on the API somehow. And we're going to see some examples today. So we have multiple endpoints. So all of them are listed in our help. So you can browse that in the help of the website. And I mean, most of them you might not use all the time, but today for the presentation, we're going to focus on one, which is UniProt KB, which is the main data set that we have at UniProt, but there are plenty of others. And also we're going to explore some tools, some like ID mapping, which is something that we provide at UniProt at EBI. But there's also Blast and Align, which is something not provided by UniProt, but provided by the EBI. And also we have peptide search, which is something provided by UniProt, but at PIR, which is another of our consortium members. And now we're going to jump to a live demo. So for the live demo, we're going to use a notebook. So in that case, this is a Colab notebook, quite similar to Jupyter Notebook if you're used to that. This is in Python, and we're going to go through some really basic Python examples, but just think that this can be applied to any other language, really, because we're just retrieving data from the internet. So this is not really limited to Python. Sorry. So you will have access to this, so no need to panic and to copy the code or anything. You will have access to that at the end of the presentation. And so I'm going to go through some basic setup. So I have some variables here to hold the URL for our two main APIs. So we have the website API that I mentioned before, and we have also another API which is more which has been around for a bit longer, and so this is what we call the Proteins API, which includes data from UniProt and also data imported from other projects into UniProt. And here I have just a helper function that will help me retrieve some of the data. So no need really to pay too much attention to that. So if you've explored the website, you will see that you can search. That's the main thing that you can do on the UniProt website. You can search for protein. And one way to do that programmatically would be to do the same thing here, UniProt KB/search with the query that you want. So here the star query means everything, and you will see that I can retrieve some data. So hopefully that works. I hope that this is the live demos. It's a bit slow. Well, anyway, so the thing I wanted to show you here, but you will be able to run that after yourself, is that I can retrieve result from that. But as I said before, you don't want to retrieve everything. Yes, the query is for everything. You don't want to retrieve everything because one thing, you're not going to be able to retrieve everything through the API. It's going to break at some point. So here you will be able to see if that works, that you can get up to 25 results in this query. And this is actually customizable, and you can modify the size of the payload that you receive, and you can customize that up to 500. And retry that. So just to tell you that those endpoints are paginated. So it means that you will have to go through different pages of results in order to retrieve all the results that you want if you really have a lot of data. So how to do that? Let's skip here. So here I have a smaller search, which is for parking and tax. So sorry, I'm searching for parking, and I'm specifying that I want that only in taxonomy ID 9606, which is human. So if one thing I wanted to show you here is that you actually have the information of how much results you get in the whole data set. So even if you do retrieve only 25 results, you will be able to know how many results you have in total. So how many pages you need to go through to retrieve that. So in that case, I have 136 results for this query. And the first page that I'm getting is, as I said before, 25. So I could change the size to retrieve 500, or I could just go through the different pages. But how to go through the different pages? We have that information in the headers of the response. So this is like the metadata around the response by the server. So in there, you can extract the URL for the next page. And you see that if I look in the headers, there's the one called link. It's just something quite standard. It's not something we invented or anything. So you will probably see that in other resources too. Is that in link, you have this link, which is listed here, that says, okay, the next page, the next bit of data will be at this URL. So you can retrieve that page, and then you will be able to go through the results. Then you do that until you don't have any next page anymore, and then you are able to access to all the data. Just going to try again to see if that works better. Okay. It's taking a while. Okay. Anyways, that's why I have this code here. So if you uncomment it, you will be able to go to loop through the different pages. So you have here this loop that loops until you don't have any next page anymore, and you're able to retrieve all the results. You can do also complex. So this was a complex search using the search endpoint, but you also have a stream endpoint, which prevents you from having to paginate through the pages. So one good thing is that you don't have to do this dance of retrieving the next page and then accessing it again. But it has some trade-offs. So one thing is a bit harder to follow the progress. So there's no way for you to know at what level of the download you are, especially if you have a big download. It's a bit harder to resume. So if you have any issue at some point, network disconnecting or anything like that, you will have to restart from the start. Whereas if you do pagination, you just restart from the page that failed. And it's not sorted by score. So by default, in the search result, you have some sorting. That means that the most important, so that would be most of the time, that would be the best annotated entries will be at the top. And so you will not get that. Also, there's a limitation of, at the moment, you cannot retrieve more than 5 million entries in there. So here, if I do the same thing as before, but replace search by stream, then we'll get just in one result. So in one query, sorry, I will get directly the 136 results that I wanted. You can also retrieve the data in different formats. So here, by default, it's in JSON. But so if I don't specify any format, I will get this information in JSON. If you want to specify a format, you could use this. So you specify the format by in the query string, and you say format XML, and you will retrieve the data in XML format. All of this is also documented on the help pages, so you're able to browse the different options that we have for the format here. You can also specify that through headers if you prefer. It depends a bit on your setup. So you can say this is standard also. It's not something that we invented. So if you say your request accepts application XML, then you will be able to get XML format. And you have some other examples here, lists. That would be the list of entries. So that would be the list of accessions. And then you have also FASTA. So if you do a search, you're able to get directly the FASTA for all the proteins that you have in your result set, and also TSV. And TSV is customizable. So one way to customize TSV is through this field query string. So if you're used to the website, you know that you can customize the columns of the search. So if you have a table of results and you prefer to display some specific column, like catalytic activity information, then you can go in the settings and change that. So this is mirroring the same behavior here. Here I'm saying I want the ID, accession, length, and catalytic activity, and in a TSV format. And this is what I will get. So if I print the result of the query, then I will get this TSV with the different information in the different columns. I think I need to then continue here. Yes.
So briefly, I'm going to now tell you, touch on how you can retrieve a specific entry. So the first box on the top left here is our main search box that you can find on our webpage homepage. I've entered an accession ID here, and this is for human parking. But in this box, you can essentially enter anything you want. You can put a gene name, a protein name, even a brief description of a function or a couple of keywords, and it will search the whole database for that information. You can also click on advanced and add different search criteria to that as well. So you might want to search for a certain gene in a certain organism, or maybe even a certain function within a certain organism, and the website will allow you to do that. But just to retrieve a certain entry of interest, I've used the accession ID here, and you just enter it into the search box and click go, and it will take you straight to the protein entry page. If you were to enter something like a protein name, it will bring you everything back that corresponds to that protein name. So then you'll be able to click on, do I want human, or do I want mouse, do I want rat, etc. So I've put the accession ID in here, and it will go straight to the protein entry page, which starts with the UniProt accession ID and protein name, protein ID, and then the protein name, taxonomy, if it's got any EC numbers associated with it, and its gene name. And then the actual entry information starts with function, and you can navigate on the far left side through the tabs for the information that you require.
And so if you want to retrieve this information but using Python, I'm going to show you another bit of code. So I'm just going to check if it started working on it again. I still have the same issue. Okay, I still have the code here, and it's already been pre-run. So hopefully this is fine. I can just show you the results without actually running it. And hopefully it was just a glitch at the moment. So what you would do is when you browse the website, you see that you have the accession here. When you browse this accession, you have this information here. This is the way to retrieve the information. So with this accession, you can query the endpoint for that, and you will do /input/060-260, and you will be able to retrieve all the information from this entry. So as you can see in this scroll bar, this is quite a lot. So some entries have really a lot of information in there, so which is quite good, but you might not be interested in everything. So what you can do is also the same way that you could customize the TSV file, is that you can also customize the payload that you retrieve from the server in the same way. You have fields here, so I'm not going to run that now because I'm not sure if it's going to work or not. And you can say, okay, I'm only interested in catalytic activity. And in that case, what I will get is then a payload which will only contain the basic information, so the primary accession, because that's quite essential to identify what we're talking about, which protein we're talking about, and the catalytic activity information. Okay. So yes. No, sorry, that's up to you, Emily.
Yeah, so I'd like to describe to you guys what sort of information we've got in UniProt, because it's probably a bit more obvious on the web page than it is through the API. So within our protein entry pages, we have loads of information and loads of different fields, but we tag each bit of information with what we call an evidence tag, and I've highlighted them here with red arrows. But essentially each tag corresponds to the information that precedes it, and it tells you where that information's come from. So basically you can go for the entire protein entry page that we've got for, say, human parking, and it will tell you exactly where every single piece of information has come from. So if you click on these tags, it will open these kind of light blue boxes that we've got here, and it will tell you where that information is from. So the blue box on the left is publication, so that's curated from that publication by curators such as myself, so that's been manually annotated. Then the alternative name down here for the protein name is an imported tag, which means it's been imported from another resource. In this case, it's HGNC, which is the Human Genome Literature Committee. And then on the right-hand side, we've got the nucleus is curated from another publication. And you've obviously got the links there to PubMed and Europe PMC for that publication if you want to read it. And then for mitochondrial outer membrane, we have a bisimilarity tag. Now that means that information has been added manually to an ortholog and therefore copied over to human. So I think in this case, I think that's the mouse ortholog of parking. And the tag here, if you click on it, expands to give you the link to the ortholog that has that manually curated data in. Next slide, please. So how this looks in the database is a series of evidence codes, or echo codes. And obviously, the more you understand about these, the better, because it means that if you specifically want to feed stuff back from a certain resource, or you only want to look at something that's been manually curated by a curator, or maybe something that's only ever been found in mouse and not in human, but it has been copied over because it's conserved in the sequence, it's pretty typical. You should be able to filter by these echo codes. So to briefly go through them, they look like the little graphics on the bottom right down here. And 269 is experimental evidence, which is added by manual curation. So this is typically from a paper. The curator has read the paper, looked at all the figures, the information, and sometimes a wider scientific field, and added that information to the protein entry. 250 is sequence similarity. So this is the example where I said the information's in mouse and is being copied over into human because it's conserved. 305 is curator inferred data. So this is where there's some information in a paper or an experiment, but it might not typically be enough to say, yes, we're sure this protein does this function. But within the field and within the proteins, maybe localization state, where it's found in the cell cycle, where maybe some of its relatives within its family are found to be transcription factors, we think this protein has a decent kind of chance of fulfilling that function. So we've added a curator inferred tag. So that's because the curator's read the field and gone, okay, maybe the paper's not got quite enough information, but it's got enough within the field to say, yeah, okay, this might be a fair bet. 312 is imported evidence. So this is from external resources. So example like the HDNC name tag that we saw earlier. On our help pages, you can find a full list of tags and why we use them, how we use them, and where it comes from on our help pages /evidences. So I'll briefly explain this because we're going to use this in the next example. When you navigate to a protein entry, that protein entry page is for the canonical amino acid sequence for that protein. So if this protein has 4 isoforms, the canonical form will be the information that's showed. If a paper or a source then gives information specific to isoform 1, a curator will then add that information and say, yeah, isoform 1 is specifically found in the nucleus, like the example here, or the cytoplasm. If a paper doesn't stipulate, we add that information to endoplasmic reticulum. If the paper doesn't stipulate which isoform, we add it to the canonical form. So in this case, there's a publication here that says parkin is found in the endoplasmic reticulum, and they've not actually specified an isoform, but they have for the other data there as well. You can find all our isoform data on the web page, just like this. This is a good example of that, actually. But you can also retrieve that information from the API.
And so to go back to the evidence code that Emily just mentioned a bit earlier, you can see that in the data, you can find it here. You will find that plenty of information in there as a evidence code with the link to where it's coming from and the ID. And those numbers are the same that Emily just explained before. Just gonna check again. Sorry about that. Okay, so to get the isoforms of an entry, what you can do is actually ask, because by default you will get only the canonical, and so in order to retrieve isoform information, you will need to opt in, and so that's what we have here. So you have the include isoform equal true here, and so if you run that for the same protein that we were showing before, you will see that you can also customize the fields. And here I say, okay, give me all the 060260 isoforms. So you will have here first line the canonical with the function, but you will see that none of the isoforms here have any specific annotated function. So here, if it would have worked, so I'm going to check if that works. I'm taking another example. Oh, it works. Okay, great. I'm taking another example. So this is P17861, and you can see that the isoform -2 here will have a different function. So this says function. So sorry, the canonical says function as a transcription factor during endoplasmic reticulum, etc. Whereas the -2 isoform says function as a stress-inducible potent transcriptional activator, etc. So you see that you can get all the annotations that are specific to isoforms in this way by including the include isoform equal true, and you will get all this information. Yes, sorry.
Sorry, I was actually looking up the answer to one of our questions that we've got. So now I'm going to show you how to retrieve and compare orthologs. So you might want to do this because you're interested in the human protein, but there's some really useful information in the mouse or the rat, or Drosophila is really sometimes a really good model for some of these kind of variants and conserved residues. And because you're interested in human parking, you might be interested in Alzheimer's or Parkinson's or neurodegenerative disorders. And looking at the orthologs and the information within our pages for the orthologs on the protein sequence can help you find interesting conserved residues, why variants affect function at times within a protein sequence, etc. So on the website, this time I've done the search by parkin, the protein name. So I've just hit search there, and you see we get a table of various different parkin and parkin-related proteins. So I've selected here, using the tick boxes on the left, the Drosophila, the human, the rat, and the mouse. Once they're ticked on the box on the left, I've just clicked add next to the basket sign in the top. And that adds the 4 entries to your basket. So we have like a shopping function on UniProt now. You can add your proteins to the basket and do different tasks and tools with them. So once they're added to the basket, you get a little basket symbol next to their entry ID. And you can run further information on them. Next slide, please. So in your basket, which is found top right of the web page, this is the view that you get on the left. So you select the entries you want to do an alignment on. So I've selected all 4 of them here, and you click align. That will compare the amino acid sequences of all 4 of those proteins and will open an align page, which I've not done a screenshot here because it essentially says, here are your 4 proteins. What do you want to call this search? I've called it parkin_alignment, and then I've just hit align, just go basically. And it will open a tool results window, which is just right at the cursor here, and it'll run the alignment, and when it's completed, it shows you a little green symbol and completed. And you click that and it'll open your results. And this tool results window is something we have for every tool that we currently have now. So you can run something, get the results, you can navigate away while it's doing a run, or you can even get the results later on, a few days later. And we keep the stuff that you've run for 7 days in your little toolbox, so you can come back to it later in the week if you need to. So the align results look something like this. What it essentially does is it aligns all the amino acid sequences and says, okay, where are these conserved amongst your orthologs? The darker the purple, the more conserved the residue. So you can see on the second row here, there's a little run of a few amino acids that are dark purple because they're the same in all the orthologs. The slightly paler purple is where it's conserved across the majority of the orthologs, and it's white where those residues aren't conserved. If there are sections of sequences that are missing from orthologs, you'll see it highlighted with a dash here. So you see the Drosophila has got a different start point to the other orthologs, and the other orthologs, the mammalian orthologs, are missing a section on the final line at the bottom compared to the Drosophila. So you might be able to use this for looking at conserved residues. Maybe that region that's really conserved amongst orthologs is interesting for you guys, or is that an active site? If it's mutated, does it cause problems in the protein function, or does it, affect disease or variants? So next slide, please. So how we look at variants on the web page, we actually have quite a graphical interface for this. So when I talk about natural variants, it's important to understand that I'm referring to the variants that naturally occur within an organism and cause issues, or that they're benign. Okay, so I'm not talking about variations in the protein or genetic code that someone has done experimentally. Okay, we call them mutations. So variants are found naturally in an organism. And on the web page, if you scroll down in Parkin, we have a whole section in disease and variants. And obviously, Parkinson's disease is a big thing for Parkin. That's what it's involved in. That's what it was named after. And we have a description that pulls from OMIM and any other additional notes that curators have added here. And this will also be tagged with all the relevant information. And then below that, we have a graphical interface that you can essentially set the variables on that allows you to filter between source, where this variant information has come from, and consequence. So is it benign? Don't we know? Does it cause the disease? So this one here, I've just clicked cause disease, so that's why it's red dots. And if you click on each spot there, that spot is a natural variant that occurs. In this case, it causes disease. And the little information box that opens here will tell you where it's from, what amino acid change occurs, if it's related to a publication, what the predicted consequence of that is, and any other clinical information. And you'll also probably find the genomic change as well. We also have another way of looking at natural variants. So at the very, very top of a protein entry page, we have a button that says Feature Viewer. Now, the Feature Viewer basically shows you from 0 to whatever the other terminus of the protein is, in a range of amino acids. Okay, so it's like a scale, start, end. And across that graphic, we plot all the regions and information we know that are specific to amino acids. So this graphic in the top right here shows you the variant section of the feature viewer opened, and all the spots relate to variants that we've mapped to that amino acid sequence. So I've clicked a variant here, 240, and it tells me the amino acid change. It tells me where that's found within a population at the frequencies. Is it important or not? In this case, its consequences are missense. It's a somatic mutation and its genomic location as well. The colors of it are the same as in the entry page. So red means likely disease and things like that. And you can also filter through, if you just want to see variants that are curated within UniProt, or do you want to find variants that are imported from resources like Cosmic and things like that. So feature view is really useful. You can look at other features, active sites, domains, etc., etc. But for this example, I wanted to show you how we show you on the web page what variants are like, and then programmatically how you can compare orthologs, find variants, and maybe compare them across orthologs as well.
And so now we're going to have a look at how to do that programmatically. So here to get the variants in through the UniProt website API, you would do well, you would need to retrieve the same kind of search that you had before. Then, sorry, let's see if that runs. So here I'm looking at Parkin search, but only for human. I'm actually limiting that to only the 5 first because that's going to be easier to show you. And so if I'm getting all the accessions, so there is format list that will give me all the accessions I'm interested in, and I print them, that will get those 5. What I can do then is ask the accessions endpoint, where you can provide a list of accessions. So that could be either through another search or something that you would have crafted manually. You have a list of accessions from somewhere, from an Excel file or whatever that you want to retrieve information for. Then you would pass it to that accessions endpoint that takes a list of accession. And as before, you can specify which fields you're interested in. So in that case, it would be variants and organism name. So just to show you some other fields, basically. In that case, we know that it's going to be human because I did the query for that. But you can see that in the results, you will have the organism name and all organism information. And then the feature I'm going to get are natural variants. So natural variants are these variants information as annotated by UniProt. There's another way to do that. There's another API that we have, which is a proteins API that will, additionally to the ones that are annotated by UniProt, will also present the ones that have been imported by from other projects. So let's try to run that and see if that works. So this is the proteins API, for which the URL you will see at the top of the file. This is /protein/API at EBI. And so if you want the variations for all the accessions I have before, you would do the same thing, request the information from that endpoint, and there you go. You will have all the information for variations on the endpoint. So it includes UniProt and also imported projects. What you can do with that is also, so as Emily said in 2 slides before about the alignment flow, is that you can actually align proteins, especially ones for which you do have annotations for, and ones you don't have annotations for, and actually try to find the similarities with them. And if you have an annotation on a specific region that is conserved, you could assume that maybe this annotation, feature, variation, or whatever might be also on this other protein that you're aligned, if this is an ortholog. So here I have the list of accession I want to align. I will retrieve the FASTA because the align tool that we use is Cluster Omega. So this is a tool provided by the EBI and for everyone to use. And what it needs is a FASTA sequence. Actually multiple sequence, obviously, if you want to align multiple sequences. And so what I'm going to do here is retrieving the FASTA. Just comment that out. I'm going to retrieve the FASTA for all those 4 proteins, and you can see that this is what I get here. And then the FASTA sequences, I could pass them to this tool, which is the align tool. So I put the documentation here. You can access it after also whenever we share the link with you. And so if you do that, you will get a job ID. So the job ID, since it might take, it might not be really, really quick. Usually it's a few seconds for most alignment, but this is something which is asynchronous. So what you need is job ID to be able to retrieve this job later. So you will get a job ID, and then you will be able to retrieve the results for this ID, for this job, sorry, a bit later. So here, if I run everything, I get the FASTA, I submit the job, then I get the job ID here, you see, and then I can I can check the status for it. So the status at the moment is running because it's been just submitted. But if I check the status again, you see it's already finished. So usually, most of the time, it's really quick. And then if I get the results for it, so if I reuse the same job ID and I get the results, you will see that I have access to the raw output of the Cluster Omega tool, which allows me to see all these alignments and those conserved regions. So combining both the annotations that you can get from our different endpoints and the result of the alignment, then you can maybe weave together all this information to try to do whatever you want with this data and analyze those aligned regions. You have also BlastFlow here that we have just put here in the code, but you will not have a look during this webinar, but you can consult afterwards.
Now we're going to move on to something that's brand new. This only went live, what, 3 weeks ago, I think it was. So this is a new project. It's called Ptm Exchange Project, and the idea is to bring in large-scale proteomics-derived post-translational modification data into UniProt. And this is done in conjunction with Peptide Atlas, Pride, and the University of Liverpool. It, at the moment, features reanalyzed phosphoproteomics data sets, and the aim of the game is to provide site-based confidence scoring for these PTMs that were brought in. I'm not going to go into great detail on this pipeline because A, we're still working on it, but B, it would take me probably a lot longer to explain this than what we've got today. I'm running a webinar in the new year, UniProt for proteomics scientists, which will have a whole section on this, and we'll also have more data by then as well. So please feel free to sign up for that when it becomes available. But essentially, the summary is we've selected good quality data sets, at the moment, phospho data sets, that we've reanalyzed and confidence scored across multiple data sets per site, so in a site-centric format, to allow users to see how confident they are and how that confidence is reflected in the strength of the evidence from these data sets that we've reanalyzed. So currently the confidence score is shown as a gold, silver, and bronze, and is based on false localization and false discovery rate. It's available to view in a modified site-based format at the moment on the protein entry page and in the API. Currently, we have rice data, so it's the data from the plant rice, with Plasmodium coming soon, fingers crossed by the end of the year, with next year Arabidopsis mouse and human. Next slide, please. So briefly, I'm going to show you how we've got the initial data in. So this is a rice protein, a translation initiation factor. And this has got quite a few modifications in this one. I'll use this one as an example. So in the PTM processing section of the protein entry page, you'll have the information from the Ptm Exchange listed as Ptm Exchange under source. You can now filter by source. So if you're looking at a protein and you just want to see UniProt manually curated data, you can select that too. But for the sake of this example, I've used the Ptm Exchange. And that Ptm Exchange data comes under modified residue large scale. So we've got filter by type there as well. We list the positions of modification. So the first one being 150. And the description tells you what the modified residue is, in this case, phosphoserine. Its confidence score in a little box. So we've got some bronze and golds there. And then the evidence tag for this being combined sources. So when you click on the evidence tag, you get this blue box that you see on the right. It's an automatic annotation because we've brought it in for a pipeline, but it's a combination of experimental data that's been reanalyzed by computational means and algorithms. So that's why that's combined sources. This particular PTM's confidence score is gold. If you want more explanation of that and our methods, the little eye icon next to the gold, click that and that will send you to our new help pages. We're going to have links to all the reanalyzed data sets once they're available in Pride. But for now, we've got source links to the original data sets in Pride and the original data, and the PubMed and Europe PMC references for those data sets. So now we're going to show you how to access that using Python.
And one thing we're going to try to reproduce is this table that you can see on the website, just straight from the data that we provide. So this is the fallback slide, just in case, but I'm going to try to do that live. So here I'm going to use a library to print a table to the results. So really, this is really specific to that library. So what I'm interested in here is for this accession, the same accession that Emily talked about before. This is the same protein in rice. And I'm going to access this specific endpoint for the protein from the proteins endpoint. This is the proteomics-ptm endpoint. And I will retrieve all the information I have about this specific accession. So here, if I print this information, I get plenty of data here. You will see all the features. And so for them, I have peptides, which are the parts of the sequence that we have information on for this specific protein. And on each peptide, I have PTMs with their position. So this is the position within the peptide, but I also have the peptide position within the protein here. It begins here, it ends here. So I can mash all the data and try to present something on the table, even though if it's not on the website, you can still do that in code. So here, what I'm going to do is I'm going to try to present a table with name, position, sources, ID, and confidence, the gold, silver, and bronze confidence that Emily just mentioned. And collect that in an array, and I'm going to present that in the terminal. And so do I have everything? So here, the first thing, I would get the PTM. So, sorry, for each feature in the data, and then for each feature, each PTM in this feature, and then for each PTM, each database reference. So that's why we have this loop star inside each other. And I will list the name of the PTM, and then find the position, which is where I need to add the position of the feature, the position of that peptide in the protein, plus the position of the PTM inside the peptide, -1 because there's overlap of the coordinates here. So I just want to take that into account. I'm going to list all the sources for that PTM, the ID of that specific Ptm, and also the confidence score, which is annotated for each of them. So if I run that, what we'll get, well, probably gonna hide the raw data. There's no point. Just gonna show the table. I will get a nice table here listing everything. So you'll see this is the same data that you get on the website. You have multiple PTMs per position here because this is coming from different studies, and you have the different scores here. So an interesting thing to do then would be, you see the positions are not really ordered here. So what I could do is order those. So what I would do is take this table data and sort by the second column. So the second column is the position. And so if I rerun that, I will get my table with all the positions ordered. Also, you might not be interested in anything that is not gold. You really want the best annotation that you can get. So in that case, you would filter out this data you're not interested in, and you would do this like this. So filtering anything that is not gold in the fifth column, this is confidence. And what you get in the end is this table here. Oops, sorry, just let it show. And then we're gonna go back to the presentation.
So really quickly, we're going to show you our Retrieve ID mapping service. Now, this is a really useful tool if you've got information coming out of a piece of software or an experimental machine, and it's using certain IDs, but you've got to take them IDs and convert them to something else, to put them in a different piece of software, maybe do some Go annotation, some pathway analysis, and your software just doesn't take what you've got out of one machine and you need to put it into another piece of software. So what we provide is an ID mapping service, and at the top of the UniProt webpage, there's an ID mapping tab. So if you click that, that opens a web page like you see here, and you can just copy and paste whatever your IDs are into this box, or you can upload a text file as well. And then there's a couple of drop-down boxes that basically ask you, what do you want to convert from? What do you want to convert it to? And there's loads of options here. This example, I've used UniProt KB accession IDs to RefSec nucleotide IDs, but you can vice versa. You can do plenty of things here, and then you can name your job. Then you just click convert, and it will give you another tools and result thing. And then when you've clicked that, when it's run, you'll just have a really, really simple table, like shown here, where you've got your original IDs that you put in and what you've converted them to. And then you can download this as a simple text file or something else as well, whatever you want to use. So this is really useful if what you're getting from one piece of software or one experiment is not what you need to put into your next set of pipeline. You can use this as a bridge, basically. And it can take large numbers of IDs as well. So we're going to show you how to do this using Python.
So this is the next example here in the collab. So here I can also, again, search this parking with taxonomy 9606. What I will get is a list of accession. I'm just going to transform that to, because it's going to be a list on new lines. I want that in a nice comma separated list here. So you can see, I don't know how many, it was like 160 or something like that. 163, I think, accessions. So you can see them here. And then if I send that to the ID mapping endpoint, which is part of our website's API endpoint, then I will be able to do a mapping from to those ID. So in that case, the from is UniProt KB accession ID. So those are UniProt accessions. So in that case, I can convert them to whatever I want. So here for this example, I'm going to use Campbell. And so I'm going to specify that I want Campbell as a target. All of those are listed on the website help, and so you can have access to all the from and tos that we support. And then I'm going to pass the list of accessions, so this list that you can see here. So I'm going to see if that runs. So if I submit that to the endpoint, so I will get, oh, right away, that was really quick. So usually I would get a job ID, and so if it was not ready, it would tell me this is running, and I would need to retry later. Since this was really quick, probably because I just run it a bit before, so it was already ready. It also redirects me automatically to the results. And here you can see the list of results. This is a simple array of from to. So in that case, this is from this accession to this accession in Campbell. And some of them, because they don't necessarily have to map all of them. You can see that the ones that don't map to anything are listed in this array. So there is no Campbell accession associated with these UniProt KB accessions. So yeah, this is the same thing because that worked. And then let's go back to the presentation. So you can see that all the code here is available for all of you to browse and to rerun. So if you go to this URL, you will have access to the code, and you will be able to run it yourself and to modify it and to save it, and just feel free to do whatever you want with that. Also, there is plenty of other code snippets. So here at the bottom, since we don't have really time, I have some examples to do some graphics with the data we get from UniProt KB. And you probably have noticed that I skipped also a blast section. So you also have a blast flow. So if you want to blast some sequences, you will be able to see that also in this notebook. So feel free to have a look at that. We're also going to share this URL in the chat and after the session.
So all that's left is to thank our funders, various National Institutes of Health bodies, EMBL, Open Targets, and SIRI. And next slide, please. And our UniProt team. So we're based across 3 sites in Hingston, Cambridge, at EMBL-EBI, in PIR in Washington, Delaware, and SIB in Geneva, Switzerland. I say our content and development team are all based across all 3 sites. Next slide, please. And we've got a lot of help and training coming up and ongoing. So on our new website, we have a whole section, if you scroll down the homepage, that shows you what webinars we've got coming up, if you need to get in contact with us, the different training at the different institutes, YouTube video sites, we have a Twitter, and we also have Facebook as well. We've got some upcoming webinars. So our next one is with Paul Denny on the 27th of October, which is a guide to UniProt for students. Now, this isn't just for students. If you just want to familiarize yourself with some of the tools and with our new website, that's the webinar to attend. We then have a webinar on functional annotation on the 17th of November, and on the 8th of December, our automatic annotation team are running a webinar as well. And as I say, coming in the new year, I'm doing a proteomics webinar, so protein tools, and I'll talk a bit more about the Ptm Exchange Project in that. If you ever need help on the website, there you go. If you ever need help on the website, we now have a little green tab on the right, and wherever you are, if you say you're on a protein page and you click help, it will give you the relevant help information for where you are at that time, or you can just contact us, and you can also search our help center as well. Up and coming at the moment is we've got our data view overall for the Ptm Exchange Project on the protein entry page, but we're also going to be bringing it into the feature viewer, both residue-centric as well as peptide-centric. The next set of Ptm data to be released is Plasmodium, hopefully by the end of the year, and our next UniProt data release is October the 12th. So now we're going to talk about we're just going to run through the questions basically, and so if anyone's got any questions to ask us or info they need, we can either answer it now or we'll pass you on and get back to you later. So I'll start. Do you want me to ask the first one?
Yeah, sure. Go ahead.
Okay. So I've used a paginated, that's how you pronounce that, right?
Yes.
Paginated, see, you can tell I don't code. I've used a paginated API based on example code you've provided, and we've not been able to retrieve results that span, say, 1,000 pages or more. It doesn't matter whether I want to retrieve full entries or just accessions. After a few hundred pages, the download just hangs. Why is that?
So this is probably a bug, so that would be good for you to contact the help. Sorry, yeah. Do you have a contact section in the website so you can contact us? And especially important thing is to tell us exactly what query you're running, because that might just be a bug for your specific query. I know I've used the paginated API for a lot of data, like multiple, like tens of millions, I think, and that worked. So it took a while. And sometimes it failed also in the middle, just because of network issues or whatever. And so the point of the paginated API really is to be able to resume. So if you fail at a specific page, you could just wait a bit and retry in a few minutes, not minutes, sorry, in a few seconds usually. And you will be able to resume from that page. and that's it. So this is the advantage. So if there's a specific issue for that, and it always fail at the same page, this is probably an issue on our side. So it would be good for us to know, and we'll pass that on to the backend team. So I'm not the one developing that, but we're all working together so we can pass that information along. Yep. I can ask you one then. I think
I've had to look both of the questions for me up.
Okay.
I will admit
So,
to this, but I do know the answers.
I will start with the one with most votes at the moment. So is there a way of linking UniProt IDs to families found on PFAM?
Yes, apparently there is. So I'm just gonna steal your screen.
Yeah, go ahead.
Okay. All right. So can you see that?
Yeah.
So I've done something really simple here just to see kind of how's best to do this, to move that bar's always right. So PFAM family, I've just taken the PFAM ID and hit that into the search on the website. And this, I've got 21,500 results, which you can filter for reviewed and unreviewed entries, organisms, taxonomy, proteins involved in X, Y, and Z. I'll show you how this information is added. So, okay, for those not familiar, that you can get this information, but this might be useful for people. But for those not familiar with this, if you put into UniProt your protein of interest, let's say, right? And this is when the website breaks when I want to show someone something. So when you get your protein entry page, raw too, apparently, you can see what data is in it and how that data is formatted by clicking on the little arrow and text. And that gives you the text file for the protein entry. So everything on the protein page is in the text file. Okay, so this is, we started off with that. Then you've got the references here and what those references are involved in. And then below that, you've got the information. Okay, so the PFAM families are automatically imported. So I've just done Control F, PFAM. So DR are the import lines, and this particular protein has got a few PFAM families. So this is why when you put the search into the web page, it pulls these entries up, okay? And these will be automatically annotated. So this will be PFAM leading this annotation. You might also find some PFAM tags for some of the domains found in here as well. If a manual if a curator's gone through and added those tags, or they found evidence for those domains as well. So you could pull that out through the API, because the API reads all these lines as well. Obviously, I've done a really rough and ready search here, but you can specifically search the domains. And as you can see, because they're automatically imported, it's actually annotated for this particular PFAM family. It's automatically annotated over 21,000 trample entries as well. So bear that in mind if you want to specify.
Can I just jump in and maybe ask you to customize the column and add the PFAM column? This is probably, yeah.
You can do this now, that's cool.
External link, sorry, the other tab, external link, just above, yeah.
Oh.
And then look for PFAM. There's so many things we're all discovering.
I didn't even know this, right? So you learn something new every day.
So you will get all your results with also PFAM in a specific column. So whenever you can,
I've just changed the column order there, look.
Yeah.
So
and so whenever that,
yeah, there you go.
And you have all of that. And if you want to access this URL programmatically, you can also hit the share button, and you will have access to copy link to the results. So that would be to copy the web view, and also generate URL for API. And so if you download that URL, you will get access. So here, generate URL for API at the bottom. You will have the way to access that through the stream endpoint or through the search endpoint with the pagination that we mentioned before.
Oh, there you go.
Yeah. With the column, with the data that you were interested in also. So that's quite good. You will need to do a TSV format. Obviously, this is a FASTA. This is selected. But if you do a TSV, you can customize the column and have that PFAM column.
Well, I learn something new every day. I genuinely didn't know I could do that. That's really cool. I hope that answers your question. It answered mine.
Okay. I guess we can So don't worry, I know it's past 2, so but we're still gonna stick around for the answer for the questions. So if you can stick around, yeah, that would be good. We're gonna answer those.
Okay, so next question. What is the advantage of running Clustal O through the web versus through the actual program? Not protein, program.
So yeah, you have actually like 3 ways of running that. That would be through the program that you host, so you install that yourself on your machine and you run that on your machine, or you can use a service like the EBI service that I've been using in the programmatic example, or you can run that through the website. So it depends a bit on what you want to do and what you're willing also to do scripting wise, because the simplest way for someone that doesn't want to touch anything close to code, that would be to run it through the website. So you have the dashboard, you can retrieve the ones that you did before. And the advantage of running that through the UniProt website is that you have the enrichment of the UniProt data. So this is not just a raw alignment that will tell you, okay, this sequence is aligned with this sequence. We will be able to annotate the chains, the sites, whatever information that UniProt has, we can overlap on top of the alignment. And you will also have the sorry, go back on the home page. And yeah, run it from there.
Okay.
Just discovered the bug yesterday, so we haven't fixed that yet. Yeah, that's.
This is when we discover bugs, is when we do webinars. It's a really useful experience. I didn't know. I'd never. There we go. I did wonder why I was doing them.
Yeah. So the same bug that's causing the error message. So you can just ignore that for now and launch the alignment.
Sorry, the Zoom box keeps getting in my way. So this is the tool results. You can see I've been really busy this week.
And so whenever that finishes, it should be 2 sequences, so it should be fairly quick. Yeah, there you go. Then you see that you will have all the annotations available through the highlight properties. So here by default, we're highlighting the chains, but you can also highlight other things. So here we're only comparing 2 UniProt reviewed sequences. So they are usually quite well annotated. So you will see both information, but that is quite interesting to compare a search with between reviewed entries, or aka SwissProt, and unreviewed entries, because then you will be able to compare those sites. Sorry, select annotation. That's yeah, I did say the other one. That is not the thing I meant.
Yeah, there we go. Yeah.
And so if you do that programmatically, you have access to the same endpoint underlying it. So you have access to that in the Collab notebook. And if you want anything like this, comparing the sites, anything like that, you will need to do the mashup yourself. So mashing up the data from UniProt, retrieved from UniProt, and the result of the alignment. So that's a bit of a trade-off. Sometimes, yeah, sometimes it depends really on what you want to do, and all 3 options are good. And the 3rd option would be to download yourself the Clustal O program and run it on your machine. That way you don't have to wait for a queue or for a job. Usually if you have quick or short alignment, that would be fairly painless to run. But if you want to run a really huge alignment and the tool just is too slow for what you want to do, you would want to consider installing that yourself on your own machine, and that way you control really the resources you give to the tool. Yep. Next question.
Yeah, I had to look this one.
So what is the best way to retrieve all proteins which undergo sulfation or sulfation? I don't know.
Sulfation.
Sulfation, okay.
Sulfation, yeah.
And the position of the sulfation in the sequence.
Yeah, so I had to look this up. So this is our rule book. So we annotate them in FT lines, which basically means we annotate them when we know the residue position, right? There's a modified residue at the position, and it'll be sulfatyrosine or sulfur, whatever. And it'll also have a keyword sulfation, okay? So what I've done here is I've searched by keyword now. Unfortunately, I mean, again, rough and ready, but to show you how you can pull this out, if you click in advanced search and keyword sulfation, and this will be added every time someone adds a sulfated residue to an entry. So that returns this list. So you can filter again on the left. I've looked through these and picked out not that one, this one. So this is fibrinogen gamma chain. And I've literally just picked this out using a keyword just to show you how this is annotated and how you can get this information. So the PTM processing section of the web page is your graphic for the amino acid chain. You can click on each of these residues, but how the sulfation is curated is it's sulfo and the residues, so sulfatyrosine. You can click on it to expand it for the sequence and for the evidence as well. So this is from a paper with the link to PubMed. So this will have been curated by a curator. So it's not automatically annotated. So how you can find this in the advanced search is you can do exactly what I've just done and use the keyword search, or you can do an advanced search again for modified residue, FT line, because FT lines are specific to the residue numbers. So far, isn't it? That's how not to do it. There you go. So this is pulling out all the protein entries with sulfatyrosine. And again, you can change your customizable columns to show that. We're going to be there. Modify residue. Yeah. Again, my box is in the one. There you go. So that's how you pull that information out on the website. You can pull modified residue information out using the API. You just, again, do the search parameters with that. You're either searching for the keyword sulfation or your modified residue sulfatyrosine, and then you just restrict to the species you're interested in, maybe certain proteins that you're interested in, etc., etc. So that's how we curate that information.
Okay. There's a couple of questions, actually comments about the nice comments about the webinar. So thank you to those comments. And a last one that just arrived recently says, kindly advise on the best way to obtain functional annotation and pathway network analysis of data analyzed using Scaffold 5.0. So I don't know about a pathway network analysis. I don't know if you know, Emily, about that.
We do certain pathway analysis using keywords if something's involved in certain pathways. So it's not what we specialize in. I will say that now. So we have molecular functions with Go annotations that are brought in. So any molecular functions and pathways that are brought in here. Keywords will have any big hitter pathways as well. We do have links to certain reactome pathways that we curate and bring in from reactome and enzyme-based curations. But from a manual annotation point of view, not so much. If it's unless it's in the function comment. With regards to Scaffold, I've not got a clue.
So just if you can just stay here on that section. So maybe if Scaffold, I'm not really sure what Scaffold provides because I really don't know about that. But here you have other pathway databases cross-references. And so just like the example with PFAM before, if you want to do a search and then highlight, have a specific column with those specific databases, because you know them or you know what they provide and interested in them, you could do the same thing. You would customize your search. So let's do maybe a search for anything like parking. Please, Emily, if you can. Yeah. And so in the same way, you could customize column. You can start customizing now, even it's not really.
Oh, can I? Cool. Yeah. You used to break the old website.
Cool. And then if you go to external link, because that would be a external cross-reference, you scroll down to there's a section about pathways, I believe. Yeah, enzyme pathways. So here you could select the databases you're interested in and display those. and you would also need to do a search that would return those specific entries. So yeah, then I'm not really sure what to look for, but yeah, you can see that some of them have annotations, and so programmatically you would be able to retrieve them also by specifying the field. Yeah, that's about it. Sorry, just need to fix that that visual bug here, but yeah.
Yeah, tiny bit. So someone's mentioned about mutual learning. This website's only gone live in July, and we are still changing it and updating it and taking into account user feedback. So please, if you love something or you hate something, love it or hate it, and let us know. And especially if something's broken or doesn't look very pretty, but please let us know about that. And just to reiterate, if you need any help, click the help tab. It'll give you some what it thinks is relevant help sections, and if not, give us a shout, contact us, and this will give you a form to fill in basically, and let us know. And also, if you're stuck and you've got questions like you're asking today, how do I do this, or what's the best way around that, or I can't get this to work, shoot us an email, you know, hit the contact form and ask us. Someone will get back in touch with you. And equally, on a protein basis, if you see something that you're working on that is obviously not been updated in a long time, let us know and we'll do it. That's not a problem.
So as you saw before, there's a lot of people working on UniProt. So even if we could not really help with that scaffold information, we have a lot of people working. We have a lot of creators and developers. So if you reach to us through this contact, then we might direct you to the person that will be able to help within our team.
Please get in touch with us if you get stuck with UniProt, or you think there's something we can improve, or definitely if something's broken, please let us know. And thank you for listening.
Thanks. Have a good day.